Now I can share. All right. So I hope everybody did have a good weekend, or at least better than mine. It was okay, but not great. Anyway, there we go. Now we're getting somewhere. Okay. Good. So what shows up there is the stuff that I want to see. And mainly just sizing everything in there. Okay. So on Friday, we talked about primary structure and a little bit about secondary structure, alpha and beta helices. And we're getting into talking about tertiary structure, the complete folding in there. We talked about fibrous proteins, which are pretty much long, linear, all sorts of stuff in there. But we're mainly going to talk about globular proteins, which are blobs. So if we were to take, say, this P50, actually this is sperm whale myoglobin in this one. If we were to take this myoglobin and just kind of like stretch out all the beta conformation, it would be really long here. Same with our alpha helix, we have a bunch here. But when we look at this in terms of its actual size, we fold it up into a small blob. All right, so we can see part of our alpha helices in here. It's a little harder to see the beta sheets because they're pretty small in there, but we have some random coil in there as well too. And of course, being myoglobin, we have a heme group in there. So we have all this, kind of folded up into this big space filling model here. It's essentially a big blob. So we're going to end up talking about that space filling model and how it folds in a little more detail. I wonder what this comes across on my video. It's like, it's actually just one, and from one side to the other there. Okay. But we'll start with our globular proteins. A lot of our folds are going to have very specific patterns to them. We can take these simple alpha helices and beta sheets and combine them to make a more complex structure. Again, the visual I always like is like taking simple Legos and building something big out of them. So we can see motifs that'll have all alpha helix or all beta sheet, or it can be a combination of those. And these motifs will be particular patterns that can be found throughout there. And our globular proteins tend to be composed of a series of motifs that then all fold in together. Okay. So what do I mean by motifs? So let's talk about a couple common ones. One is going to be our beta alpha beta loop. So in this case here we're looking at parallel beta sheets. How do we connect the end of one beta sheet to a region of another? Well, we can have this alpha helix that loops around there and allows you to make that parallel beta sheet connection. So this beta alpha beta loop is very common and just a good connecting unit in there. We can also see for things where we might have pores that need to penetrate a membrane, we'll make a beta barrels. So we essentially have a bunch of beta sheets here, and not super easy to see, but we have anti-parallel beta sheets here that are kind of woven around to make this big tube or barrel shape in there. Okay. How do we kind of put all these together to make something? Maybe, there we go. Whoops, too far. Too far. All right. We can take multiple beta alpha beta loops and kind of combine them together to make this particular fold. All right. So here we have several beta sheets that are arranged in here, and it's almost like we're making a little handful bowl shape structure in there. All right. So we end up making a combination alpha beta barrel. All right. So it's artistic. Okay. I'll say sometimes takes a little getting used to looking at these ribbon structures and to looking at our space filling structures. one of the things I actually greatly enjoyed about my graduate work was doing the molecular modeling, because we'd actually have like the funky glasses and special monitors where you could have a 3D view and actually be inside the molecule. So but that, you know, just with practice, you can visualize these and kind of see where the connections are. I don't talk about that as much, but that's something that just kind of comes with practice to kind of help determine where you are in the molecule and what different parts are close to each other. Okay. So besides these highly ordered alpha helices and beta sheets, we have proteins that can be intrinsically disordered. Okay. So, you know, when they're made of a lot of random coil, or they may have multiple conformations that work as well. So we'll have some segments that lack definable structure. So we might see, you know, lots of prolines in here, or I would say maybe some glycines because of their highly high freedom of rotation around the feed and phi angles. So these disordered regions can conform to a lot of different proteins, and basically allows them to be adaptable and interact with other stuff, with a, you know, large number of possible binding partners. Okay, so we can actually talk about Ponder, P-O-N-D-R, which is a prediction of natural disordered regions based on our primary sequence. And the closer this is this scale, the Ponder scale ranges from 0 to 1. Closer to 1 it is basically 100% of disordered. Closer to 0, the more likely it's in a particular pattern. So in this case, we're looking at a P53 molecule here. And the center region, kind of in this, you know, beige or salmon color, I guess that's really not beige, pinkish, whatever color this shows up as, is fairly ordered. There's like one little section here that's got some disorder to it, but basically everything is below this kind of 50% line. So we've got a tightly organized structure in here. Our amino terminus and carboxy terminus are both much more disordered. There's a lot more disorder in there. There's a few sections that are kind of locked in, so there may be a few critical motifs in those sections. You can see maybe this alpha helice here near the C terminus might be that particular thing that's in there. But we have this bunch of disorder over here. And one of the neat things about the P53 molecule is, depending on, you know, which major molecule it's a component of, you can see very different structures in there. You can look at the carboxy terminus with a bunch of different components on here. Maybe we're talking about cyclin A or sirtuin or CBP bromodomain or the S100 housekeeping protein in there. So we'll have some different structures here, and that will allow this to interact with a variety of different binding partners shown here in the different colors. Okay? So we can, you know, change, you know, adapt our order to interact with these particular binding partners. So that disordered region can, you know, depending on the role of this P53 protein, can interact with different binding partners in different parts of the cell, different times of the cell life. Okay. So what's the takeaway from this? What we're looking at here is that we have an overall structure, an optimal fold, but some of that fold is variable and some of it is disordered. Okay, so our role of our protein is in part determined by its structure. Okay. All right. And we'll touch back on that here coming up in chapter 5. Yes, yeah. Probably not. No. At most, you would say, you know, if I gave you a scale there, you'd say, well, is this going to be more likely to be disordered, or is it more likely going to have a particular shape that it's in? So, yeah. And that's about all we'll do with that. So, all right. So we've talked about primary structure, which is just the sequence. Secondary structure are alpha, beta helices. Tertiary structure, which may incorporate motifs, but is basically the entire three-dimensional shape of one particular peptide. We also have our quaternary structure. Right? And our quaternary structure is if we have multiple subunits, like in this case we have hemoglobin, we have these 4 hemoglobin subunits, usually 2 alphas and 2 betas, and the 4 of them kind of come together to make this overall structure. So if we have more than one peptide as part of our protein, then we will have a quaternary structure. This is our large functional cluster here. So we could compare, and we'll do this in chapter 5, myoglobin to hemoglobin. Myoglobin just has the one bit, the one, what I want to say about that, the one peptide, and our hemoglobin has the 4 peptide units. So myoglobin would have primary, secondary, tertiary structure. Hemoglobin would have primary, secondary, tertiary, and quaternary. Okay. All right. Just a second here. Okay. Alrighty. And so if we move forward, okay, how do we actually figure out these structures? Maybe we'll move forward. Come on. There we go. Okay. All right. We got 2 primary methods that we utilize, and we have X-ray crystallography and NMR. Okay, and they both have advantages and disadvantages. So we'll kind of briefly talk about these 2, and you can almost imagine an exam question compare and contrast, X-ray crystallography versus NMR. So what do we need to do for X-ray crystallography? We've got to purify our protein. Okay, that's good. That's not too bad. We got to crystallize the protein. All right, that for some proteins that's easy. For many proteins, particularly ones that are membrane bound or mostly hydrophobic, that can be really challenging. Then we need to collect the diffraction data, calculate the electron density, and fit the amino acid residues into the density. The good news is there's no size limits. We can have massive proteins and work with this. It's very well established. The con is, membrane proteins make it really, really hard, and you can't really resolve the hydrogens in there. Yeah.
So on the NMR, you can see the hydrogens, but that's when you do the N plus.
Yes. Yeah. Yep. And you'll actually see this in about 2 slides. We'll see that in there. So if we look at our diffraction pattern, you got this nice little diffraction pattern there. You get this density. You can find your major structures, and then you can figure out probably where the hydrogen should go. Every once in a while, there might be like, where does this particular hydrogen go? That can be helpful, yeah. But for X-ray crystallography, this diffraction pattern will give you all the information. And again, the advantage is you have a huge, you can do massive molecules in here. There's no size limit. As opposed to NMR, the more molecules you have, the more complex that spectra gets, and the harder it is to assign peaks. The big difficulty, though, is crystallization. So, for example, I looked at the Rieske iron sulfur protein. The workaround, because this is primarily membrane bound protein with regions that stick out into the intercellular space and the intermitochondrial space. They used antibody fragments that were soluble and attached that to the purified protein complex. And because the antibody fragments were huge and soluble, they were able to get that to crystallize. So that was a strategy for dealing with membrane-bound proteins.
So overall, is this easier to get the structure that you're looking for than NMR? Because NMR, you have to do like data reduction and other steps.
So the answer is it depends. If you've got a really huge protein that you can make a crystal of, I love this method. It's the best. If you've got a small protein, like maybe cytochrome C or something that's fairly small, and something that or it's something that's relatively small and hydrophobic that you can't make a crystal of, because crystallization is not easy. I mean, I was very glad that we had collaborators who did the crystallization for us, because a couple years later I tried to crystallize some stuff and I was like, this, it requires a great deal of training and experience to get good crystals. But if you got something huge, this is great. On the other hand, for NMR, you've got, again, something smaller, something that you can't crystallize. Come on. All right, let's do it this way. Okay. For NMR, you got to purify the protein, so no worries. You got to dissolve the protein. So, okay, that's usually not too bad. Collect the NMR data, assign the signals, and calculate the structure. The good news is you don't need to crystallize the protein, because, like I say, for many proteins, crystallization is ridiculously hard. And with NMR, you can see most, if not all, the hydrogens, or at least a lot of them. Doesn't work too well for insoluble proteins. So if you can't dissolve it, it doesn't work out too well. And the bigger your protein, the more peaks you have, the harder it gets to assign them or to differentiate them. So if you look at typical NMR data, what we're looking at here, this is a globin, probably a myoglobin in here. So you end up with some lovely data there, and you can assign these peaks in here, and you can actually kind of resolve many of the hydrogens that are in there. So, yeah. All right. So if you have like a massive protein, I'd probably say, let's try to crystallize it. If you've got a small protein, we'll go NMR. If you can't make a crystal, probably have to go NMR. Okay. So not a whole lot on there, but those are two primary methods, the two methods there. All right. So what you may have noticed in those previous slides is that a lot of times our protein folding is actually fairly complex. You look, well, that one's not too complex. It's great. We use a smaller protein for the X-ray crystallography example there. But there's a lot of folding that goes on there. So the question we have to ask is, well, how do we actually get to that mature peptide that's properly folded? So we can kind of look at the life cycle of a protein. Right? So if we go back to our mRNA and the ribosomes, we start making a nascent polypeptide in there. Here's our sequence in here. We have this nice little folding intermediate. Sometimes we can just fold correctly right off the bat. We get directly to our native protein. Sometimes we may require chaperone proteins to help it fold. Okay? And we'll see some examples of both. Every once in a while, something doesn't fold properly. So maybe you can unfold and try again, unfold and try again. Or sometimes something goes wrong and we get aggregation. Okay? So we might have these big blobs, or we might have oligomers that show up from our misfolded proteins. And if you think of prion disease, things like Kuru or Creutzfeldt-Jakob's or mad cow disease, these are diseases that result from misfolded proteins, and you end up with these beta amyloid plaques that form in the brain, and there's not a good way to deal with them because there's not a good scavenger in there. So you basically just get holes in your brain till you go insane. So there's never mind. Wasn't that a Cypress Hill song? No, okay. But these plaques are all from misfolded proteins. There are some misfoldings that can be accounted for and corrected. What you might see is misfolded proteins get ubiquitinated, and then they'll go to a ubiquisome that will cleave them into small fragments, and maybe we can try again. All right. But, we get our native protein here. Eventually, our native protein, all proteins have a lifespan. Eventually, even if they're correctly folded, they'll be tagged with ubiquitin and destroyed as well. A lot of times, our misfolded proteins that aggregate will also be attacked and chopped up into small pieces as well, too. So we focus on, I tend to focus on prion disease because, I grew up on a cattle farm. And it's one of those crazy brain diseases, and it is prion diseases are relatively unique in that they are the result of a misfolded protein. So fold something the wrong way and you die. All right. Okay. So folding is important because our 3D structure is going to determine its function, right? So if we lose our structural integrity, then we can lose that function. We have loss of function as well too. So basically if we unfold our protein, we call that denaturization, right? And we can denature proteins in a lot of different ways. We can change temperature, we can change the pH. So again, you know, we'll be either adding hydrogens or abstracting hydrogens. So we're going to be adding and changing charges, so that can cause it to unfold. We can stick things in various solvents. We can use chaotropic agents like high concentrations of urea or guanidinium hydrochloride will also cause them to unfold. Let's take a look at some examples of this. All right. So if we look at ribonuclease A, right? If we start heating up ribonuclease A, it's stable. You know, get the body temperature is very, very stable. Get up to about 50, 55 degrees, and all of a sudden, it just unfolds. Like it's the temperature that broke the ribonuclease's back. Right? So it goes all at once here. This very steep curve basically means it's pretty much good. Then you have this critical threshold where it unfolds. And this would be our melting point where half of it is unfolded. Okay? If you compare this to apomyoglobin, you know, we can see that it's stable up until about 70 degrees, but you know, it starts unfolding somewhere around 55, 60. So it starts unfolding in the same region, but it's a little more stable over a wider temperature range there. But still, as we heat it up, we cause it to unfold. We'll see this again in lab when we talk about PCR, our polymerase chain reaction. We utilize a heat-stable polymerase that basically we can heat it up and it won't denature. So it's stable at high temperatures. So basically these polymerases were isolated from bacteria that grow in volcanic vents. So the temperature there is like 80, 90 degrees Celsius. So they have polymerases that are stable at those temperatures. Okay. If we look at our guanidinium hydrochloride, we can look at our concentrations here. 1 molar, we're fine. 2 molar, we're fine. 3 molar, it's like, we get to about 3.5 molar, and we start denaturing our ribonuclease. Okay? So we're basically just kind of intercalating our guanidinium ions into the protein, which is disrupting the hydrogen bonds and eventually causing it to fall apart. Okay? All right. So here's the cool thing. We got ribonuclease in both of these, right? So there was a very simple experiment, incredibly simple, and it won a Nobel Prize. So the idea here is ribonuclease is a really small protein. We got these 8 cysteines that form disulfide bonds. If we add urea and beta-mercaptoethanol, it's going to denature it. So our beta-mercaptoethanol is necessary to break those disulfide bonds, and the urea is the chaotropic agent that disrupts the hydrogen bonding. Okay? So if we take our purified ribonuclease, it's folded into its native fold. We add urea and beta-mercaptoethanol, and it falls apart. Okay? So then we dialyze it. Undergo dialysis, and we remove the urea and the beta-mercaptoethanol, and it'll spontaneously refold back to its native shape. So what this means is that the sequence of ribonuclease alone determines its native conformation. Right? So the sequence information contains the folding information as well. Okay? And this was done in 1972, so not terribly long ago. So biochemistry is relatively new in terms of field here. If we look at this visually, here's our ribonuclease, and here are our disulfide bonds. We break those bonds, add our urea, and we basically get a denatured protein. We dialyze away the urea and beta-mercaptoethanol, and it goes right back to its original form. Okay. So we don't need anything else. In this case, ribonuclease is small enough that it doesn't require chaperones. We'll get to that level of detail here in just a second. But our sequence determines our structure, and our structure determines our function. So sequence data controls functional data. All right. So how do they actually fold? You guys ever try origami? It's like, look, a swan. Yeah. Made lots of, look, it's a big ball of paper. It's a joke. Okay. So we can actually follow this in sequence, okay? So there's a distinct path that proteins will take. So we might see this particular peptide, we're gonna see this little range in here kind of fold in a particular way and then form a beta sheet. And then these beta sheets, actually we're gonna see these other regions do this as well too, form a beta sheet. And then the center region is gonna make an alpha helix, which will attach to this beta sheet and then it'll kind of fold in here. So we basically have this kind of 1, 2, 3, 4 or 5 step process with a couple steps happening simultaneously, okay? Right? And they, just snap together and get into that shape, okay? Maybe it's like one of those fancy lawn chairs with a table on top and a little side thing there. You unfold it and you have your drink stand and laptop stand and you sit back there and maybe your feet thing kind of kick up, but it just kind of folds in there nicely. You'll see them go across social media every now and then as well too, but there are a lot of like little tables or stands that will unfold to hold all sorts of things, or fancy the way they fancy folded furniture. I don't know what you'd really call them, but something like that, where they just kind of snap into place, and then you can spin it the other way, and they all snap back into the way that they want to be. Where there was like a kid's toy, like this little buckyball thing, where you could expand it out. It makes this big sphere, and then you can wad it up into a small sphere. So that's kind of like what's happening here. We're folding in a particular sequence to get there. The question that we then have to ask is, how can proteins fold as quickly as they do? Okay, they're going to go to their lowest energy fold, all right? So they do this, in at most a few seconds, all right? So we're looking at microsecond to second time scale. So if we're just doing this randomly to find the lowest energy state, I'm going to go back to thermodynamics here, right? You guys ready for some thermodynamics equations? No, we're not going to do that. But we're gonna just theoretically talk in terms of thermodynamics. We want to go to the lowest energy state. So our lowest energy state is going to be our stable conformation. Okay. So if we try out all the possible combinations, because if you think, maybe we have 100 amino acids, so we're looking at maybe say 1,000 atoms in there that all have different orientations they can have. It would take years, if not millennia, to find the most stable. So this is our Leventhal's paradox. We find this low energy state much, much faster than random chance, right? So clearly it's Jesus. But what we have here is a non-random direction towards that native state, okay? So there is a method to it. I like this next slide just because of its pictorial representation. What we see here is probably what we, most commonly imagine our folding to be like. So our bottom of our funnel here is our lowest energy state. That's where we want to get to. So we have this funnel here. We basically have, a bunch of pathways that will ultimately lead us down to the most stable outcome. All right. It would be great if we were like part C, where there's just one way we can do it. There's only one way. All right. So we just immediately kind of get there, but that doesn't happen. This is probably a little more accurate, where we have a bunch of stable intermediates. Okay. So if I go back up to that picture here, that'd be kind of like saying, well, this is fairly stable. Maybe we want to hang out here for a little bit before we fold over this way. Or maybe our little alpha beta loop here. Actually, this is an alpha beta loop, whatever. Maybe this is more stable and it takes a second to get down here. It pauses. All right, it's like when you're taking a trip and stop at a rest stop, or you'll see the world's largest ball of twine or something. So those would be a series of local minima that you kind of have to overcome to get down to the very bottom in here. Okay. We can also have a case like in D, where we have a significantly stable intermediate. And this may contribute to misfolding, because maybe we get stuck in this trough and we can't find our way over this energy hump to get down here. So it's like, yeah, I planned on going to Kentucky, but I stopped in Tennessee and it was so nice I decided to stay. That's a terrible analogy there, right? We kind of get stuck here. So this could contribute to misfolding, where we kind of get trapped in this intermediate energy state. Chaperone proteins can help prevent that. to get them either over the hump there, or to take them out of the that little energy minima. Okay, so the more complex our proteins, the greater the possibility that we can have some of those somewhat stable intermediate states. Okay, so if we take our protein, we get to the folding intermediate, we can make a mistake, have a big aggregation in there, and shoot it all up. We get to our native protein. But sometimes, if there's a chance of misfolding, we'll use these chaperone proteins in here to help hold things in the right position to make sure it doesn't misfold. It would be kind of like somebody directing traffic, and it's like when we get the hard rainstorm and that road closed, got to go around this way. It's essentially what the role of our chaperone proteins. We're binding our nascent protein and making sure it doesn't get trapped in one of those local energy minimas. Okay. If we look at, some chaperone proteins tend to be huge and might require energy in here. So they might be driven by ATP hydrolysis. So if we have like a big protein, we can actually kind of insert part of the protein in here. It'll interact with our chaperone protein, and we'll eventually get to our native fold in here. I'm not going to go into excruciating detail about how that works. Just what I want you guys to know is that chaperone proteins have binding regions for the proteins that they need to help fold. You can kind of see they come in here maybe in some non-native fold, and by interaction with the chaperone protein, it puts them in the right shape. So it's kind of like the step-by-step guide to origami. Look, it's this one. You follow this step-by-step, or if someone helps you do that, you get from your piece of paper to your swan. I need to change that example. I always say swan. Got to find something else you can do with origami as well. All right. So that's the way that folding works. I mentioned this a little bit earlier, but if we have misfolding, we can get a bunch of diseases in here. So a correctly folded beta amyloid is a soluble globular protein. All right. So that plays a role in neural cells. But if we misfold it, then what we end up is with these beta sheets that are partially exposed, and then the beta sheets from different molecules can associate with each other. They can actually have that stable hydrogen bond interaction and start stacking on top of each other. So what you get is, instead of this nice little soluble globular protein, you start seeing lots and lots of these beta amyloids coming together to make a kind of semi-fibrous protein, and you end up with this beta amyloid plaque that's insoluble, and that can cause pretty severe damage in here. So we actually end up losing our helices. Our helices maybe kind of unwind or something. Something goes wrong with them, but we have a bunch of these stacks in here, and it just propagates. And so you encounter another protein, and it's more stable to suddenly bind with all these. It's like mob rule, and was it dilution of responsibility? All the other proteins are unfolding. You should do it too. Yeah, you ever have your parents saying it? Well, your friends were jumping off a bridge. Would you do it? There you go. So for some of our beta amyloid plaques, the answer is yes. That's bad. All right. Good times, right? So this is our significant chapter in here. We're talking about structure, talking about alpha helices, beta sheets, talking about tertiary structure there, properties of fibrous and globular proteins, how we actually determine the overall structure, and how proteins fold. I should say that that folding is still not terribly well understood for a large number of proteins. I know every year there's contests to predict the folding of a protein using a variety of computer models. So it's getting better and better, but we still can't plug in a primary sequence and say, it's going to look like this. You kind of go back to like the whole Star Trek thing. It's a strange new protein, and project it up there, and you get this three-dimensional structure. Can't do that yet. So maybe for some proteins, for relatively simple proteins, but that's an ongoing issue that is being worked on. So basically what I'm saying is, there's lots of stuff where I'll say, we don't know this yet. We don't know the details. So there's lots of career opportunities in biochemistry. You can be a professor at a small college. don't know if that's a good thing or a bad thing. Not too bad. Pay could be better. Ooh, let's put that on. Yeah, send this to the dean. Dr. Ebert needs a raise. Okay. Terrible. Okay. All right. So what I want to do now is actually, this chapter segues very nicely into chapter 5. I think on Wednesday, I'll kind of review the exam, because I did finally get the exams graded and final grades posted on over the weekend sometime. But I'll go over the exam on Wednesday. But I want to go ahead and jump into our next chapter now, chapter 5. All right. Oh, come on. Let's do this. There we go. Okay. I really need my multi-monitor set up, here and here. Okie dokie. Okay. So chapter 4 is all about protein structure. Chapter 5, all about protein function. Okay. And they are, intimately tied together. Okay. So what we're going to talk about in here, and you're like, oh crap, we got a bunch of bullet points here now, right? Terrible. We're going to talk about how proteins will bind with their various binding partners, our ligands. We reinforce this when we get to chapter 6, because we'll talk about enzymes and their substrates, which is very similar to proteins and ligands. Just we're going to do stuff to the substrates. We'll look at some modeling of these interactions. And then we're going to use hemoglobin and myoglobin as canonical examples to look at oxygen binding in there as well too. And we'll talk about regulation of oxygen binding. And then in the last part, we're going to talk a little bit about antibodies and antigens, how they interact. We'll talk a little bit about actin, myosin, and muscle contraction, and then how we regulate that as well too. There's actually this kind of neat little power stroke. And I need to find there's a very nice model, a three-dimensional microscopic model of myosin walking along an actin filament. So we'll see that. All right. Okay. So for protein function, what we want to be able to do is to interact with a binding partner and then release it. So we have to have reversible binding, right? So I'm going to bind something and I'm going to release it. Okay, it may be as simple as carrying fuel from the outside of the cell to the inside. Right? How do I get that across the membrane? Maybe I can bind to a protein, take it inside, and release it in there. So we'll look at the specificity of ligands for their various binding sites. Right? We can talk about how they bind, and our ligand binding is going to be coupled to conformational changes in the protein itself. So we can call this an induced fit. We'll talk about that in comparison to a lock and key model, where like your key fits in the keyhole there. That would be perfect binding, but then it's not easy to release that. So we have this induced fit model that we talk about. We'll also look at how sometimes some proteins can bind more than one of the same ligand. So maybe if one binds, it makes it easier for the next site to bind one. So we have this idea of cooperativity. So the different sites can influence each other as to whether they're going to bind the ligand or release it. Right. And we'll see this illustrated by hemoglobin. We'll talk about antibodies a bit and muscle proteins just a little bit, because this is a long section. The exam 2 material, we got 3 chapters in here. It's long, it's rough, but this is the worst section. Yeah, not this chapter, next chapter, but there's a lot here. Okay. So I try and not make it horrible. All right, so let's talk about our globular proteins. A lot of chapter 4 we talked about, the fibrous proteins and how they're, like our keratin, how they make the different fibers, and the fibers, microfibers wrap around each other, and so on and so forth. So our globular proteins do a lot. They can store stuff. We can look at myoglobin storing oxygen in the muscles, ferritin storing iron within the cells. We can look at transport as well too. Hemoglobin transports oxygen from the lungs to the tissues. We can look at serotonin transporter in the brain as well. We can use defense against pathogens, various antibodies that form, your various cytokines for whenever there's a particular health insult. We can look at muscle contraction. You know, I was up here, swing, it's awesome. You know, pointer, love this. Much better than a laser pointer because I can whack things. But in order to do that, I had to have actin and myosin and muscle contraction going on here. And again, we'll talk about catalysis, and we'll save most of this for chapter 6, but we can look at biological catalysis and enzyme activity on here. So our globular proteins do a lot, all right? So let's look at this, you know, go back to basic chemistry and look at this in terms of a chemical reaction. So we have a reversible transient process of chemical equilibrium. Oh, I'm saying the bad words. Dynamic steady state. But we're going to look at this kind of in isolation here, where we can have, you know, maybe a protein and its ligand, and they come together and form a complex. So it's basically A plus B gives you AB, and this is reversible, right? So if we have a molecule that binds to a protein, we're going to call that our ligand. Usually it's a small molecule, but if you think of something like DNA binding proteins, you have small proteins that are binding to massive molecules in there. But usually we're going to think it's the small molecules. The region where it specifically binds is called the binding site. So if you think of, you know, like the room you're sitting in, you know, in this case, if you're in the classroom, you got lots of chairs, you've got your chair, that's your binding site, right? Because everybody always sits in pretty much the same chair each time, right? Yeah. No need for assigning seating, we assign it right? The ligands will bind via the non-covalent interactions that, you know, govern the overall structure as well too. So we're looking at hydrogen bonds. Again, for when you have senior seminar, you know, somebody's going to ask a question, the answer is going to be hydrogen bonds. This is it. This is our non-covalent interactions. They're going to fit in the right spot here. Might not necessarily be hydrogen bonds. It might be hydrophobic interaction as well too. But the fact that this is non-covalent allows these interactions to be easily reversible. Okay. So this is what we got so far. Right? Not too bad, I hope. Okay. So let's look at this pictorially. We can draw our little Pac-Man symbol up here. Yeah, that sound getting so old, my people might not know what that is anymore. But here's our protein. Here's our ligand. They bind together. We get a protein-ligand complex. Right? We can have an association constant. We can have a dissociation constant. Okay. So our association rate constant Ka, dissociation rate constant Kb. One in each direction there. Right? When we hit equilibrium, what that basically means is that, you know, the rate of ligand binding is the same as ligand dissociating. Okay? So we could actually write up a nice little rate complex here and say our Ka of our protein concentration, ligand concentration is going to be equal to our Kd of our protein ligand complex. This is at equilibrium. Right? So now we can rearrange this and make it maybe a little easier to work with. Okay? So we can look at this large Ka here. This will be our equilibrium association constant. Okay? Capital Ka. We could invert that and have our equilibrium dissociation constant Kb, but mostly we'll talk about this Ka. Right? And if we see that, this is going to be our concentration of bound protein and ligand divided by our reactants, concentration protein, concentration of ligand. Okay. So basically Ka equals little Ka divided by little Kd. Okay. And you flip that for your Kd here. But we'll talk about our association constant, okay? And we'll see that a lot. All right. So if we make a solution that has a certain amount of protein and a certain amount of ligand in that, it's pretty easy to figure out what our concentration of protein is, pretty easy to figure out what our concentration of ligand is, but it's almost impossible for most partners to figure out what's the concentration of bound ligand. So that's a little difficult, all right? So what we're going to look at is our fraction of bound sites, okay? So if this is our Ka, here's our same equation, okay? We can actually, this should be little Ka, right? Yeah, we can say PL is basically just Ka times concentration P times concentration of L. So we can actually get rid of this, okay? And then we can rearrange this equation, right? So if I substitute that in for there, we can rearrange this and give us our equilibrium dissociation constant. Ooh, yeah. So how we do this, let's look at this. Our theta is going to be PL over PL plus P. All right. So put in our KAs here. It's our fraction of binding sites. So basically this is occupied protein. This is total protein on the bottom here. Okay. Okay. So since we can't measure this, we'll put in our Ka and our LP. Okay. All right. So if we do that for both of these, get that going on here. Okay. Now I'm going to start isolating. You can see that our concentration of protein is in all three of these terms. So I can cancel that out. All right. So I've got Ka times L over KAL plus 1. If I rearrange a little bit more, allergic to math. Okay. If I divide everything by Ka, I can basically get my concentration of ligand over my concentration of ligand plus 1 over Ka, or I can call that my dissociation constant as well too. Right. So my fraction of occupied binding sites is going to have this equation. Right. So see if you can follow along that. Here's my fraction. Occupied protein, total protein. I do some rearranging. I can basically get rid of my concentration of protein. What this means is that my fraction of occupied sites doesn't depend on my concentration of protein. Okay? It only depends on my concentration of ligand. Right? So let's try an analogy. If I look at this room,
so when you cross out, again, you start from that, I guess you cross out everything in front of the plus. They're all the same on top of the bottom.
Yes, but because you have this over here, you really only have, so there should probably be another step in here to make it a little clearer. Crank this up here. What we should probably have is kind of an intermediate in here, so we'll go this way and then back down to here. My theta is Ka times L over K, whoops, brain and fingers not talking. Ka times L plus 1. Okay, so now.
So you cancel out P.
Yes, cancel out the P from everywhere. And then what I'm going to do is I'm going to divide every little section by 1 over Ka. That gives me this. So my Ka will cancel out here, but I'll have 1 over Ka there. And then I can just call that Kd, my dissociation constant. So when I get rid of the P, I'm left with 1 down here. Okay? So now I want to collect my terms. I want to get rid of Ka. So it'll cancel out here and here, but I still need to account for it over here. So that's where I get just my ligand by itself and 1 over Ka for this 1. So I'm essentially on the top, taking Ka, L over Ka, and then on the bottom, I'm taking Ka, L plus 1 divided by Ka. So I can have that cancel out there, but I'll still have 1 over Ka left over here. And again, just running through the math there, the important thing that tells us is our fraction of occupied sites only depends on the concentration of ligand. So if I increase ligand concentration, I'm going to fill up more of the protein. The analogy I like is like this room probably has about 35, 36 chairs in it. So theoretically, we could have 36 chairs occupied. If we have like 2 students come in, we're not going to occupy a huge fraction. If I have 4 students come in, it's more likely we're going to occupy more of those chairs. If I have 20 students come in, we're going to have a greater fraction occupied. If I compare that with the big lecture hall downstairs that has 70 chairs, if I still have 2 students come in, I'm still gonna, there's still gonna be that greater likelihood of more chairs being occupied if I have 20 students than just 2, no matter the size of the room. So our fraction of occupied binding sites only depends on the concentration of the ligand. Okay. All right. So let's look at this visually. All right. Graphical analysis here. The fraction of bound sites depends on the free ligand concentration and on our KD, okay, our dissociation constant, because that's going to play a role. But that's a constant for each protein, okay? So experimentally, we can, basically graph different ligand concentrations and see what's occupied there. And then, we can plot this nice little hyperbolic curve and figure out our KD, okay? So we can either do that graphically or these squares. So the neat thing is, when KD happens to be equal to the ligand concentration, half of our sites are going to be occupied. All right. So if this equals L, we'll basically have L over 2L. That's 1/2. So that's our 50% occupancy. We can figure that out. Okay. All right. Is this the best way to do that? Not really. We could actually plot this a different way to generate a straight line and a line equation and figure out where this is. But we see our asymptote up here, total occupancy. In practice, we pretty much almost never have complete occupancy. Okay, just thermodynamically not going to happen. Okay, let's look at a specific example. We'll look at oxygen binding to myoglobin. Okay, so here we have our cartoon of our myoglobin structure with our heme in the center. We're going to have oxygen bind right in here. All right. In this case, rather than looking at concentrations, because oxygen is a gas, we're going to look at partial pressures. Okay. So instead of having our dissociation constant, we'll have our partial pressure constant for oxygen. Basically, 50% of it's going to be off there. So our P50, basically. All right. We can look at the higher the pressure of oxygen, the more sites are going to be occupied on myoglobin. And we end up with this awesomely great curve here, okay? Basically, myoglobin is going to bind oxygen at very low concentrations and hold on to it. Okay? So you don't need very much oxygen from myoglobin to bind it. Okay. All right. I don't know if you guys ever read like the text for the company, the slides in there as well too, but you get lots of information there too. They're talking about the picture and how there's an artifact in here. And anyway, I should point out too, I forget, but we can divide up these different regions here. And here's like our helix A, our helix B, and then here's our connection, our AB connection, disordered coil, and then B goes to C, and C goes to D, so there's a CD. Presumably there's a BC back here that we can't see. D connecting to E, E to F, F to G, G to H, and our carboxy terminus. So just ways to kind of look at that. So we can say, oh, we're talking about the E barrel, or the A barrel is, and the A helix is far away from the E. The E helix actually contributes this histidine that helps stabilize it. But that's more than you need to know right now. All right, but again, looking at this binding curve, the greater the concentration or partial pressure of oxygen, the more is going to bind. Okay. Okay, so thermodynamically, all right, we can look at our interaction strength and associate this with our association constant, and or the inverse of our dissociation constant. Okay, so we can compare this to free energy. Look at free energy. We have enthalpy and our entropy in there. Okay, we have our Ka basically equals something like this. Our Kd is the inverse of that. So we can look at another delta G relationship, negative RT natural log of our Ka, or RT natural log of our Kd, whichever in there. Okay? So what this comes down to is if we have very strong binding, okay, our dissociation constant will be very small. Okay? So the smaller our Kd, the more tightly our ligand binds. Okay? So strong binding is just kind of arbitrarily defined as a Kd of less than 10 nanomolar. Okay? Weak binding is going to be defined as greater than 10 micromolar, okay, for it to bind, for our KD constant. And then in that 3 orders of magnitude in between would be intermediate binding. Okay, so basically the smaller the value for KD, the stronger the binding. Okay, so we can look at a couple examples in here. Our typical receptor-ligand interactions are going to be in this range in here. Sequence-specific DNA, we see that we're in kind of like the nanomolar range, so that's pretty strong binding. Same way with our antibody for antigen, pretty strong binding. Our enzyme substrate, actually kind of weak, which seems a little surprising initially, but we'll talk more about that in chapter 6 and why that is that way. If you look at something that's got super strong binding, biotin, avitin. So if you look at like biotin, avitin purification cartridges or columns that use those, you want to pull your protein off of there, you will pull it off really tightly, and you might have hard trouble getting it off of the beads or the stationary phase. That is a super high affinity, very strong binding on there. Okay. All right. Check that just to make sure. Okay. Too many random messages on here. All right. So let's talk about specificity and how people thought about it historically, and then ways that our initial models have been improved. Okay. So a lot of proteins typically have really high specificity. They're only going to bind with particular ligands. So we might have like an NADH binding molecules, or we'll see ATP binds to a lot of things, but but maybe we'll have glucose binding molecules, or concavalin A, which binds mannose. This very high specificity can be explained by complementarity of the binding site with a ligand. Okay, they fit together. Our chairs are shaped the way they are so that your butt will fit in there. Okay, you guys know about industrial engineering, McDonald's seats, and the whole interior design of McDonald's. They're initially comfortable. You kind of go in there and like, oh yeah, you sit down here. But the shape of the benches, if you sit in a McDonald's bench for a long period of time, you start to ache a little bit, right? They're designed to be initially comfortable, but to become uncomfortable over time. So you want to come in and sit down, but then you're like, it's uncomfortable, so now I want to leave. So the next person can come in and sit down. So you can get this constant flow of people in and out, so that you make more money that way. Same thing applies to the color scheme. It's these bright, inviting colors that want you to come into Mickey D's, and then after a while they kind of glare and get on your nerves, so you want to leave. So there's a whole series of studies to see what appeals to people. All right. So you might think of like the McDonald's table benches as being relatively complementary, but not perfectly complementary. It's not like the Lazy Boy, or the adjustable pressure in there, so that you can memory foam, so that you can sink down into it and just go to sleep there or something. That would be. So when we talk about complementarity, we mean that the size matches up, the shape matches up, charges will match up. Maybe one is positively charged, one's negatively charged, so they attract. Or our hydrophobic and hydrophilic characters match up. We might have two hydrophobic protein, hydrophobic ligand, or hydrophilic, where we'll have some charge based in there. Got to bring out my most awesome. It's the world as an example here. So here's our protein, here's our ligand, and they kind of fit together nicely. So if we go back, 125 years, you've got Fisher, who proposed this lock and key model. That basically our protein was like a keyhole, and our ligand was like the key that fit in it. Okay. Just perfectly match up. And if you have the right key, it'll fit in there, and you can turn the lock and open the door and all that good stuff in there. But it turns out there's a thermodynamic problem with that. But what happened is, if your key fits in there and it fits perfectly, it's going to be energetically favorable to stay there. So the lock and key model kind of fails because it doesn't lead to a transient interaction. It's a more stable interaction, and you'd have long-term binding. So a better model is this induced fit. Okay? So what we have here is our general shapes of the two are about the same, but you can see these two pieces aren't exactly perfectly complementary. So what happens as they get closer together, the shapes of both change just a little bit so that they make better binding partners, okay? So this requires, you know, a change in the shape. So we're going to a higher energy state for that shape, but that's overcome by the attraction that's in here. But we want to go back to our original shape, so that's going to make these kind of fall off and resume their original shape in there. So this is our induced fit model. It's like we're close, but we're not perfect. All right. I want to make analogies. I don't know. I don't know if I'm brave enough to make it. I got tenure, but who knows here? Yeah. But, I debate. Only because we're recording. My words can be used against me. Okay. So here we'd have maybe something like this shape. Our ball doesn't quite fit in there, but as we get closer, you know, we're going to see this induced fit model. Okay, this is not the optimal shape for this. So I want to kind of go back to my triangular shape, which is going to squeeze the ligand back off. So we can see induced fit, all right? And that's going to encourage, you know, ligand binding and ligand release. Okay, all right. The other neat thing about induced fit is that I can bind more than one ligand now. So if I've got, you know, like molecule A here, I can have this induced fit, have molecule B that's got a rounded shape in there. I can also maybe bind it. And maybe I prefer this one, so I might have a higher specificity for, you know, ligand A as opposed to ligand B. All right. Awesome, right? You guys are like, sweet. Okay, good. Look at my time here. I have to keep checking my schedule. Like, what time do we meet? You know, what time do we end? All right, we got a few more minutes. Okay, so we'll introduce our first case study here. Okay, and I just want to start on this just a little bit. Okay, not go too far into it. We're going to look at the globins. And in particular, we're going to compare myoglobin and hemoglobin. All right, so we have this problem. Protein side chains don't bind oxygen very well. They're composed of lots of oxygens, but they can't bind it. We could use transition metals, but then we get a lot of free radicals. Free radicals are bad, okay? They'll cause lots of damage. If you get that reference, you're really old. So what we end up doing is taking organometallic compounds like heme. They're more suitable. Problem is, we don't want to oxidize iron in free heme to iron 3 because we get a very reactive form there. So we come up with a solution. We have a heme that is protein bound. So we're kind of sequestering this very reactive iron 3 species away. We're also making sure that we're not going to get a lot of radical formation in there. So we're kind of protecting the overall organic structure while still being able to bind oxygen in there. Okay? So this protein bound heme. And our globins are our heme binding proteins. Okay? All right, so myoglobin is going to be the example of our storage protein. Myoglobin is where we store our oxygen. Hemoglobin is where we transport our oxygen. Okay? Both of them, very similar structures. It's just hemoglobin is essentially 4 myoglobins stuck together. All right? So let's look at our porphyrin ring. This is our porphyrin ring, which is the basis of our heme. All right? So this central conjugated ring structure here allows these nitrogens to coordinate with iron. Iron has 6 coordination sites. So these 4 planar ones, the nitrogens in the porphyrin ring, coordinate that way. And then that leaves binding site behind the plane and in front of the plane there. And we have our various hemes. We'll have these various tail groups that come off the porphyrin ring that define what they are. Okay. Space filling model. We've got a lovely iron in there. I guess that's supposed to be orange to distinguish it from the oxygens up there. And we can see our coordination in here. We've got this planar coordination and then 2 other coordination sites for that iron in there. And so what happens is we insert our heme into our globin molecule, and we'll have this histidine residue with this nitrogen that will take up the 5th coordination site. So here's our planar porphyrin ring coming in from one side. We have a histidine, and then we have this open slot here where oxygen can bind. All right? And this is going to be on the interior of our globin. So we can stick our oxygen in there and it can bind safely We don't have to worry about radical formation. We don't have to worry about highly reactive iron 3 species or anything crazy like that going on in there. Okay, there we go. So basically, it's like those little chests where you store stuff. You put it in there and deep in the center and hide it away from the world. So that's what we're doing with our oxygen. Okay. All right. So let's take a look at this. Carbon monoxide, is about the same size as oxygen, and our heme will actually bind carbon monoxide 20,000 times better than oxygen. Okay, and that's because of the filled lone electron pair that can be donated in there on the iron 2. So one of the ways we overcome this, okay, is the protein pocket is actually going to have some steric interference so that we can't fit that CO molecule in there as well. So it's going to reduce that by about 2 orders of magnitude. But still, carbon monoxide is going to bind 250 times better than oxygen in your body. So what that means, you may have a carbon monoxide detector in your home. You don't want to definitely want to park in a garage with your car running in the winter to warm it up, because carbon monoxide is going to compete with oxygen. If we think of the atmosphere, fortunately we have way more oxygen than we do carbon monoxide, so it's not something we typically need to see. But if you're in an enclosed space with, like, say, running, you see this after a lot of storms where people lose power and they have a generator, maybe like run it inside the house, and they get a buildup of carbon dioxide and die. That happens after pretty much every loss of power or something like that. So that's why you have to be careful, because carbon dioxide is going to bind. And when carbon dioxide binds, it binds much more strongly than oxygen, so it's going to outcompete it. And the reason for that is because of the shape in here. Our oxygen is going to bind this way. Okay? Our carbon monoxide is going to bind this way, and you can see we have this nice linear interaction between all the p orbitals that are in there. So our binding pocket is actually going to have this distal histidine in here that's going to inhibit carbon monoxide from fitting into the cleft where it can bind. So that's what's dropping that from 20,000 to 250 times. Okay. All right, so question for y'all now as we wrap up this little section here. How do you treat carbon monoxide poisoning? Anybody know? So if you got somebody who inhales a bunch of carbon monoxide, well, you can give them oxygen. All right, so if high concentration of oxygen will eventually outcompete. So if you've got 250 times as much oxygen as you do carbon monoxide, it's about 50-50. You're going to have about half down there. If you got 10,000, so basically super high concentrations of oxygen will eventually outcompete the carbon monoxide. So that's how you can do that. And that relies on the fact that these are transient interactions, okay? So there's another interaction that can occur here as well too, which I don't have on this slide, but cyanide, CN, can also fit in here really, really nicely, okay? It does, right? But there's a difference. Both of these are ligands that will come off. When cyanide pops in here, it makes a covalent attachment, okay? So how do you treat cyanide poisoning? Yeah, no, you guys are like, hey, are there people online too? I'll let one of them answer now. Well, you can't outcompete it. You can't give someone oxygen because the cyanide is permanently bound there. So there are a couple strategies for dealing with cyanide poisoning, and that's why cyanide is so deadly and so fast acting. So this is where I'm going to offer everybody almond cookies at the end of class. But it basically binds permanently. So you need to give something that will bind any free cyanide that hasn't attached yet, so you can get that out of there. Blood transfusion might work to put in new hemoglobin. So there are some strategies for that, but it is much more time intensive. It's like you basically can't have a first responder treat cyanide poisoning very well. You need some pretty serious hospital equipment. Generally, usually, especially blood transfusions fairly easy there too. But you need something to bind to scavenge any unbound cyanide as well too, because basically that cyanide is going to essentially kill whatever globins have it bound. So our hemoglobins is going to basically prevent oxygen from getting to the rest of the body. Okay. All right. So that's a good space to end. We'll pick up on Wednesday. The first thing I'll do on Wednesday is talk about the exam and kind of go over that a little bit. Generally, I was fairly pleased. I don't have any issues with the exam. Scores were generally pretty good. They were where I wanted to be. I think the average was like 77 or something. I forget. I had to look again. But most of the class was in an area where I felt comfortable with. There's like one or two students I'll probably send an email out to and say, hey, we're working on stuff there too. But for the most part, people were doing fairly well. I was a little nervous because I graded my freshman physical science class, and oh my God, they were awful. The online stuff is not working too well with them. But you guys being juniors and seniors for the most part, your exam data is like within less than 1 percentage point of historical data for the class. So we're doing okay here. I got to change some of my approaches with my freshmen, but you guys are all right. So if you did well, if you were in like the 80s or better, life is good. Keep doing what you're doing. If you're in like the 60s or 70s, we'll strategize and find ways to make sure you get stuff there. And if you were one of the few people who were lower than that, well, we can talk about ways we can get you on track. Okay. Yes. That was the average test.
Okay.
The bad news is this one coming up.
I mean, it doesn't seem too bad so far.
Yes. Yeah. Chapter 6 is all about enzyme kinetics, and that's where that's where people get scared.
Is that math?
Yes, but we're not going to do a whole lot of math in there.
Okay.
But it's a lot of graphical interpretation and in keeping track of, well, wait, what's a competitive inhibitor versus a non-competitive inhibitor versus it's just keeping track of a lot of stuff. And it's complex. And there are 3 chapters in here. After this, like I say, exam 2, worst exam. Historically, that's where everybody does badly. So don't sweat that too much, because the 3rd exam is like a 20-point swing in points. I've actually had like the class average on exam 3 be 88 before. So exam 3 is the easiest exam generally. Yeah, I mean, well, there's 2 more after that, 4 and 5. They're fairly average. They're like this first exam, except you know what I'm talking about. You know the way I'm going to be asking questions, so they're usually not too bad. And like I say, after this exam, it's only 2 chapters for each of the remaining exams. So I think part of this is just a little bit long, but our protein section is just 4 chapters long, so can't break it up too much more. Okay. All right, I will let you guys all go. I should stop my share and stop my record, and I'll post this as soon as I get it back.